It is time to talk about proteins, and I believe these are quite important as well. We talked about already nucleic acids, and these have important roles in your cells, but proteins have a lot of functions that we're going to discuss later on in this tutorial that are needed for you to be alive. That's why your cells need proteins, and that's why you classify these molecules as biological molecules. For chemists, they usually prefer to use organic molecules because these molecules are comprised of a lot of carbon atoms. But for molecular cellular biology purposes, we're going to classify them as biological molecules. So the second thing that I would like to talk about is that they are linear, non-branching polypeptide chains. What do I mean by this? Well, these proteins have monomers, and the monomers are amino acids, which you can also abbreviate with AA. You will see that in textbooks, so keep that in mind that we're talking about amino acids. And these proteins are comprised of a lot of amino acids as monomers, and they are bound to each other through peptide bonds. Okay, and when you have two or more amino acids, for example, I will say at least two, you call it peptide. This is a peptide, okay? And what I mean with this is that they're linear because this will form a line. These amino acids bound to each other, they form a line from A to B. If you stretch this line, it can be arranged in different forms, and you're going to see that later. But if you stretch that line, it goes from A to B. It's a line, like a necklace of amino acids, let's say. And with non-branching, I mean that as branches of the trees, you will not see branches of other amino acids chains or peptide chains coming out of these of this main line. So this will not happen. So there are non-branching, and this is what I mean with non-branching polypeptide chains, meaning that proteins are comprised of several chains of several molecules like these here, several polypeptides, meaning several peptides, okay? And these peptides, but the peptides, if you stretch them, they'll always end up with the line shape, okay? That's what I wanted to say to start with the proteins. And the second thing, the last thing, sorry, that I would like to mention is that, look at this picture here. You have here an extraction, protein extraction of several proteins that have been extracted, I'm assuming, from a live tissue. And they look like this to the naked eye. That's quite nice, but you can see here that they are macromolecules. So macromolecules that, even though you see them here, they're mixed here and they look, there is no defined molecular shape here. All you see is just a pellet there of gathering of proteins that ended up with this shape. But this is what you can see with the naked eye. But they are macromolecules, that meaning that if you compare to other molecules that you know, these are some of the largest ones. When we look at proteins and look at their functions, it's a great way to actually understand what these molecules are able to do, or the importance of these molecules in your cells. And if you go a little bit back and look at DNA, which is not a protein, is a nucleic acid, but you can look at DNA, which carries genetic information, and that genetic information, once converted or expressed, it is expressed into proteins. And these proteins have functions in cells. And these are the functions that we're going to look at. And once we do that, you're going to have a better idea of what I'm talking about. Whenever I talk about proteins, you can have an idea, okay, these macromolecules are involved in these functions in my cells, okay? So The first function I want to talk about is structural, which means that proteins serve as structural components of cells and tissues. And one example I want to talk about quickly is cytoskeleton proteins. And these proteins, like actin and myosin, they form this very complex meshwork in the cell's cytoplasm, which helps in not only the structure of the cell, the shape of the cell, but also the transport of some molecules throughout the cytoplasm. So it's a very important structural system, and it is comprised of a lot of proteins. And so keep that in mind, structural. The second one is regulatory, which means that there are some proteins like transcription factors that are involved in regulation of certain pathways in the cell. And an example that I'm giving here, transcription factors, these proteins are actually able to bind to certain DNA sequences, and in this way they are able to control some gene expression. But this is a very simplistic way to describe a transcription factor, but we will learn more about them. You need to learn more about them, especially when you talk about gene expression. The third function is transport, and the name says it all, but I can give you an example of a protein that functions in transport, and this is important, and we're going to talk about it when we talk when we discuss the structure of the nucleus of the cell. And this molecule here, this protein, actually moves other protein molecules into the nucleus by binding to a specific recognition sequence. But just have an idea that these proteins that are involved in transport, they're usually in membranes, embedded in some membranes, and they help bring in or out some molecules. But keep in mind, this is an example of a transport protein. The fourth function that I have here is signaling, which means that proteins are involved in information that is transmitted between cells. Okay, this is a very simple way to look at signaling, and proteins are definitely involved. And I can give you two examples: polypeptide hormones, which are secreted into the bloodstream and have endocrine functions, for example. And the other one that I want to mention is adapter proteins, which are actually accessory to main protein in a signal transduction pathway, which we will talk about because signal transduction is very important, especially when you talk about cancer. But keep in mind, just understand that they are signaling proteins, and that's another function that you find in proteins, in these types of molecules. Another one is defense, and an example of this is antibodies, which are proteins that help fight infection. You probably heard about them. And another one is stress proteins, which according to the stress found in the cell or in the environment, these proteins are able to trigger and help change some features of the cell that we're going to learn later, which help to cope with the stress, the changes, the sudden changes in the environment. This is what we talk about when we talk about stress. But right now, just keep in mind that they are able to also have important roles in defense. The other function that I want to mention is receptor. They can be receptor or they can be receptor proteins. An example of this is hormone receptors. And these are usually on the surface of a cell or in its interior, and these receptors are able to bind to specific hormones and trigger some pathways in the cells, but proteins are able to be the receptors for these hormones and other molecules as well. Another one, the last one that I want to talk about, last function, and it's also a class of or a subtype, let's say, of proteins, are enzymes, and we are going to talk about enzymes a little bit more in detail at the end of this tutorial. But what I want to say is that these are proteins that catalyze reactions, and we're going to see how they catalyze reactions. But for now, just have an idea that proteins can also have this function. We know that proteins, I mentioned previously, that proteins are comprised of monomers. Okay, they're polymers, and polymers have these building blocks called monomers. And monomers, the monomers found in proteins, the building blocks, are amino acids. And this is the abbreviation I explained previously, which you're going to see from now on whenever I mention the protein's monomer amino acid. And I have a structure here. This is the basic structure for an amino acid. We're going to analyze it. And I also want to tell you that there are 20 different amino acids. Okay, so there are 20 different amino acids out there, or in your cells or in your body. You have 20 different amino acids, but they all have a same basic structure, and this is the basic structure they have in common, and we're going to have a look. So the central, in my center, let's say, it's a central carbon. So this is a carbon atom in the center. On one side, you have a carboxyl group, 1 carbon and 2 oxygens. So this is a carboxyl group. And on the other side, you have a amino group. And this is common to all 20 amino acids, including this hydrogen atom that you see here. This is also common in all 20 amino acids, this structure. The thing that differs from one amino acid to another in this list of 20 is this R group here, also known as side chain. Side chains actually define the role or determine the role of the amino acid. So I'm going to write here, determines or role of amino acid. This is going to be different in each amino acid, and you're going to see when I show you a list of all amino acids, and you're going to see that they have all these 4 structures in common: the carbon, the hydrogen, the carboxyl group, and amino group, but the side chain is different from one another. So now if you still look at the amino acid structure that I just explained previously, you see that here you have 4 structures: the central carbon, the hydrogen, the amino group, and the carboxyl group, that are present in all 20 amino acids. But there is this side chain here, represented as an R. This is what I could call the ID card, or the fingerprint of my amino acid. They're all going to have a different side chain group. And these side chains actually have functional categories, meaning that they have different roles. They play different roles. And I'm going to mention 4 main functional categories here to exemplify, or to give an example of what I'm talking about this fingerprint here, the difference between all these amino acids. So the first one I'm going to talk about is basic amino acids, which means that the side chain here is going to be basic at physiological pH, meaning that it's going to be positive. So we're going to have there on the side chain of basic amino acids a side chain that is positively charged at regular physiological pH. The second one is acidic amino acids, which means, oh, and let me tell you that examples of that are arginine and lysine. And we're going to look at the list of the actual drawings, the images of all these amino acids, so we can get a better perspective, a clearer perspective of what I'm talking about right now. The acidic amino acids means that at regular or physiological pH, I'm going to have a negative charge on my side chain, so negative or negatively charged, okay? And you can say that these are, some people call these polar amino acids. And why are they polar? Because they clearly have a marked positive or negative side chain, a clear charge here, which make them polar, okay? The third functional category that I want to talk about is uncharged polar amino acids, which means that there is no charge, no actual positive or negative charge, but they are, the side chain is actually polar, which means that there is a molecular configuration or some atoms present in the side chain, which gives some polarity. And with polarity means that if you have a polar molecule, it's going to be able to interact with water, especially if you try to dissolve it. So meaning that it's hydrophilic molecule or hydrophilic side chain. The nonpolar amino acids, the last functional category that I'm going to talk about, means that there is no charge. And if there is no charge, in this case, the molecules will have a configuration that will not allow polarity. So there is no polarity. And if you have a question on polarity, I would definitely suggest you to go and look at your chemistry notes because you really need to have it down. But polarity meaning that you will have a negative and positive poles, but not exactly an exposed charge. Because these molecules usually have some sort of configuration where one atom is going to, in a way, pull more, due to electronegativity, pull some more electrons to one side, and therefore you're going to have a side of the molecule that is going to be somewhat more negative than the other side. So one negative side and one positive side, and that's what polarity in a nutshell is. But please go over your chemistry notes. And no polarity means that these molecules or these side chains are going to be hydrophobic, means that they do not interact well with water. They do not like water, let's say. So I want to make the point that I just explained previously using the actual images of amino acids, so you can clearly understand by looking at the structure, the actual structure of what I'm talking about when I talk about functional categories. And the first thing that I want to do is talk about basic amino acids and acidic amino acids, which are polar. They have polar side chains. And for basic amino acids, like I explained previously, they have polar side chains with positively charged side chains, let's say. And if you look at these three, for example, I'm not going to go over the 20 amino acids. I'm going to go over the main ones so you can get a general idea. So if I go over arginine, histidine, and lysine and look at the structure, this looks a little bit different than what I'm used to explain or draw. But I have here my central carbon. My H should be somewhere around here or there, or depending on the configuration of the molecule. Here I have my amino group, here my carboxyl group, and here I have the side chain, or the R, and I'm going to name R1. In histidine, I have the same thing, my C amino group, my carboxyl group, and my side chain, which I will name R2. Lysine, the same thing, my carbon, amino group, carboxyl group, and my side chain. And you can see common things, or this structure in this, what I just wrote in these structures, in these side chains. They're definitely polar, they have a positive charge, and they're when you find in normal physiological pH. So they're basic amino acids. These are examples of what I just talked about. With acidic amino acids, I have negatively charged, so they're still polar, and they're negatively charged. They have negatively charged side chains. Same thing if I look at aspartic acid or aspartate. I have my amino group here, my carboxyl group here, and I have my side chain here, which I will name R4. If I look at glutamic acid or glutamate, I have my carbon in the center, amino group, carboxyl group here, and my side chain here. And same features that I just mentioned. I'm going to do R5. And same features that I mentioned here, that they're polar and then negatively charged. You can see here that these side chains are charged, and therefore they're acidic because they are found like this in normal physiological pH. Now it's time to look at the uncharged polar amino acids, and like I mentioned previously, they are uncharged, so no charge, and I'm going to use this as my example. So my C center and my amino group here, carboxyl group here, and then I have my side chain here. I'm going to name R1 on this one. Same thing with threonine, and I have my C amino carboxyl group and side chain R2, and asparagine, C amino carboxyl, side chain R3, and the same thing here. I don't want to be repetitive, so you get the point at this time, and the R4 here. But what I want to tell you here is that they are uncharged, so no charge. And by no charge, if you look closely at these side chains, you don't see any charge here, do you? No charge, like you saw on the previous polar amino acids that I just showed you, the basic and the acidic. And you don't see a clear exposed charge here, but these molecules have some sort of configuration here. For example, if I look at this R1 here, that has a hydroxyl group, and the hydroxyl group for itself does not have an exposed charge. Like, you don't see a negative charge or a positive charge exposed there. This is a very simplistic way of explaining, because if you go into chemistry in more detail, you're going to have, this is not the right words to use. But what I want to say here is that you can see that even though you don't have a clear charge here, an exposed atom with a charge, you do have more electrons going this way in the oxygen, towards the oxygen, and therefore you're going to be left with a negative charge here and a negative positive charge on the side of the hydrogen. That's more or less what they mean. There is no clear charge, but there are poles. There is a negative and a positive pole, so they are polar. And the same thing applies to the other three. If you look, they have molecular configurations where the electrons are going to be going toward a certain area of the molecule, and therefore they're going to be left with some areas with more electrons where are going to be more negative, and some areas with less electrons that are going to be more positive. Now the nonpolar amino acids, these side chains, like I explained previously, they have no charge. And trust me, I'm not going to go over all of these because you are going to be very bored and fall asleep on top of your computer. I want to keep it simple. But if you look at isoleucine and look at the side chain here, let's say R1, you can clearly see that there is no charge, no exposed charge, and no polarity. So no polarity, and with no polarity, I mean that if you look previously when I explained the hydroxyl group, that my oxygen is definitely more electronegative than, this is basic chemistry, more electronegative than my hydrogen, and therefore my electrons, are going to go in this direction, and therefore I'm going to have a negative charge here and a positive charge here. You don't see that here. The electrons are distributed in an equal manner. That's a very simple way to say it, but in a way that there is no positive or negative charge, no poles here. That's why there is no polarity. And for this reason, these side chains are going to be hydrophobic, which means they do not like water at all. They do not interact with water very well. And this is what we see in these side chains. If you go in this one that I love to pronounce, phenylalanine, which, ooh, I did well. Well, if you look at my side chain here, I'm going to call it R2. It's a benzene ring, which, if you learn from organic chemistry, these are definitely hydrophobic because they're non-polar. The electrons are so well distributed in this molecule that there is no actual charge, positive charge on one side and negative charge on the other. It's just, well, they're well distributed. The charges are well distributed in this molecule. So I want to talk about peptide bonds because when you go from one amino acid binding to another amino acid and binding to a third and fourth and so on and so forth, you are forming a peptide. So two or more amino acids together bound to one another, you are forming peptides. Okay, these are peptides. And polypeptide chains together form proteins. Okay, so definitely, like I mentioned before, my amino acid is going to be, the amino acid is the monomer for the proteins. But they have to bind to one another, and they do it through these bonds called peptide bonds. And they're here. These are the bonds that I'm going to talk about now. And if I look here, I have, let's call it my first amino acid here. And this is my second amino acid, so 1 and 2. And you can see here, if you look closely, this is my central carbon, and then my hydroxyl group, my hydrogen, my carboxyl group here, my hydrogen, my amino group, and my side chain. And if you look at my second amino acid, I see my central carbon, my carboxyl group here on this side, a hydrogen, an amino group here, and a side chain as well. So you can clearly see the structures here of the amino acids. And what's going to happen is that the bond is going to be formed between the carboxyl group of my first amino acid and the amino group of my second amino acid. So the first amino acid is going to give up one oxygen, and my second amino acid is going to give up 2 hydrogen from the amino group to give 1 water molecule. So 1 water molecule is produced in the peptide bond. And if you go and see here, I'd even decided to put the same color. So this is my first amino acid here, and this is my second amino acid, and the bond is formed right here in the middle between the carboxyl group of number 1 and the amino group of my second one. So this is my peptide bond. This is my peptide bond. Another thing I would like to explain is that we have two amino acids here bound to one another, but imagine you want to add a third one and a fourth and so on and so forth, and you continue this chain. So if you look here, the direction is going to go from this direction, and you're going to see that it's going to always, the peptide bonds are going to be formed with one carboxyl group of one amino acid with the amino group of the other. And what's going to happen is that you always call that the ends of the polypeptide chain, of this chain, if you look at this end on this side here, so say this end here is called a N-terminus. You need to know this nomenclature because we're going to talk about later. So the N-terminus, because you have an amino group, the N from the amino group. And on this one, as the name also indicates, very simple, because of the C of the carboxyl group is C-terminus. Terminus, okay? So keep that in mind. Every time we talk about N and C-terminus, you know what I'm talking about. It's the ends, we're talking about the ends of the polypeptide chains.